Okay, hello everyone. Thank you for being here, for the whole lot of you. But I'll make it worthwhile and show you something extremely interesting. We're a small Finnish biotech, but you need to understand that we are founded and guided by the world's leading immunologists. We are people that discovered the first adhesion molecules back in the 80s. We told the world some 15 years ago, what does CD73 do, now a hot topic in IO. We told the world that, hey guys, maybe you should use Herceptin for gastric cancer. And now we're with this novel targeting Clever-1. This is our pipeline in a flash. So we have two clinical programs, the CD73 program, which we call Traumakine, and then our anti-Clever program, which we call Clevergen. We also have an upcoming clinical program that's not disclosed yet. Today's topic are M2 macrophages, tumor-associated macrophages, and converting them and putting on your immunity. This is the competitive landscape. These are all the targets out there that have to do with myeloid cells. I just need to highlight that none of these targets are M2 specific. Macrophage targets have been notoriously difficult because macrophages are difficult. They're so plastic. But you have to understand, yes, the T cells will do the killing, but the macrophages will command the T cells. This is the basic concept. Well, I'm going to show you some very interesting, exciting data on how we activate T cells even though we target macrophages. Now, macrophages digest and present to T cells should this be attacked or protected, and they secrete pro-inflammatory cytokines. Clever-1 is a big scavenger receptor on macrophages. The thing with the Clever positive macrophage is that whatever it digests, it doesn't present. So instead of presenting the tumor to the immune system, it hides the tumor away. This phenomenon is also seen in pregnancy as the placenta is filled with these Clever positive macrophages. You have to think of a Clever positive macrophage. It's a subpopulation of M2s. Not all M2s even express it. But we see this as the most immunosuppressive population. And you want to change these, and you want these to start presenting the tumor to the immune system. The thing we found is that whether you're Clever high or Clever low expressing truly dictates, are you responsive to current IO therapies and how well do you do. If you're Clever high expressing, in other words, your macrophages hide what's digested, you do not respond and you die. If you're Clever low, you may respond to current therapies and you survive in cancer. Cancer types we're targeting have the same phenomenon. If you're Clever high, you usually die. If you're Clever low, you survive better. This is mice data. Clever knockout mice do not grow tumors. They don't get metastasis, not in lymphoma, not in breast cancer, not in lung cancer. So the knockout data is absolutely beautiful. And the reason being, we do the macrophages, but the T cells now in the left-hand corner, they infiltrate the tumor, and tumors do not stand a chance. Same results are achieved with a mouse anti-Clever antibody. The mouse antibody is a surrogate antibody, as Clever-1 and Clever-1 in mice and human are not identical. Anyway, we put anti-Clever-1 treatment against anti-PD-1 treatment in lung cancer, even outperformed the current market leader in a single agent therapy. Moving on to clinical data. We're currently running this large basket trial in Europe with leading European sites and KOLs. We are now moving to the US side as we have our IND approved there. We have now completed the dosing of 30 patients, so the phase 1. We spent more time in finding the right dose, as we see that the immune system is activated very well with almost all doses. This seems to be a very potent activator of the immune system. Now we're currently moving on to the expansion cohorts. First clinical signs have been seen in colorectal cancer, so ovarian cancer, and that's where we're heading first. We as a small biotech need to think carefully where we put our bets. So this is the clinical data in man. So you know companies target NK cells, Tregs, CD8, CD4s, but we get them all. We upregulate NK cells, CD4 cells, CD8s, we even downregulate Tregs. We also increase B cells, which is a new, very interesting topic in IO. We were looking to get even more clinical responses judging from the cell responses. But again, we're treating end stage, very advanced cancer patients. In this slide, I want to highlight that the CD4, CD8 cells we activate there are Th1 type, not Th2 type. And these cells secrete interferon gamma. Now that's what you need if you want an effective IO treatment. Again, advanced late-stage patients. It's very hard to win the game when there's been 7 lines of chemo and you don't have a working immune system. And we lost a lot of good candidates just because of brain met swelling, closed compartment, they can't take it, they need steroids. Steroids dampen and block the drug effect. That's known in IO. This is the extremely interesting part. I have seen nothing like this, and talking to a lot of people, nobody else. This is now T cell populations and exhaustion markers on the T cells before and after treatment with anti-Clever-1. Basically, even though we target the macrophages, exhaustion markers on T cells, and I may say all of them, they are down-regulated. There is no single agent like this that can do all checkpoints. As I say, one ring to rule them all. So to highlight again the basic concept, we do the macrophages, but the macrophages will do the immune system. There's nothing like this out there. IP coverage is all us, and there's a biological basis for a targeted population, as they have to have these clever positive tumor-associated macrophages. Again, being a small biotech, but there's a lot of prestigious institutions and names behind this work. And any questions?